Good morning and good afternoon to the people on the other side of the world. I am Rutvi Vaja, a third year undergraduate in biomedical sciences and research. My research areas are specifically neurobiology, and I like to investigate a lot related to neurodegenerative disorders. Amidst the COVID pandemic, our wet lab research had no access because of the lockdown and everything. So I tried to involve myself into bioinformatics and dry lab research. So I wanted to always apply the bioinformatics techniques in order to solve complex problems related to neurodegenerative disorders. So this project was done for 8-month long fellowship program. The title of this project is in silico analysis and affected biological pathways in multiple sclerosis. Just to begin with, multiple sclerosis is a neurodegenerative disorder that is characterized by demyelination of neurons. It is also regarded as an autoimmunity disorder in which the T cells are supposed to be attacking the nervous system and especially the myelin sheath of the nerves. And the myelin sheath is responsible for maintaining the electrical insulation between the brain and the body, also responsible for maintaining the kind of signals that the brain passes to the body. In case of multiple sclerosis, since the patients are very much affected due to demyelination of neurons, they lose the contact between the body and the brain. Furthermore, they also suffer from tremor, seizures. Epileptic seizures are very common. And then multiple sclerosis progresses from stage 1 to stage 4. There are 4 types of multiple sclerosis. When we look at the multiple sclerosis in depth, earlier it was regarded as a white matter degenerative disorder. But since last 10 years, research have shown that not only white matter, but it also have started affecting the gray matters. So the entire idea behind my project was to investigate the formation of gray matter lesions. Why are they formed in patients with multiple sclerosis? Because originally it was regarded as a white matter degenerative disorder. So in order to brief you all about my research questions and hypotheses, in order to target my main central question, I performed the 3 main hypotheses. The first hypothesis being, I wanted to find out the affected biological pathways in multiple sclerotic patients by comparing it with the control samples. I also wanted to find out the association of multiple sclerosis with T cells, because since multiple sclerosis is regarded as an autoimmune disorder, CD4 and CD8 cells attack the myelin sheath. But how do they attack? What is the association? Why do they get overreactive is what I wanted to investigate. And my third and the final hypothesis that I wanted to investigate was I wanted to find out what are the upregulated or the downregulated genes in affected biological pathways of multiple sclerotic patients. So to start with my study, I took a data set from NCBI Geo. That data set consisted of 184 total RNA-seq samples, out of which 105 were the samples of multiple sclerotic patients without any gray matter lesions. 37 were samples of patients of multiple sclerosis with gray matter lesions, and rest 42 were the control samples, which were used for comparing and contrasting. Talking about the methodology of my study, it was an RNA-seq analysis study, and hence I divided it into 2 parts. First one was exploratory analysis, in which I wanted to analyze all the 3 groups. And the second was comparative analysis, which is a sort of downstream analysis. For comparative analysis, what I did was I did a t-test between normal appearing gray matter patients, gray matter lesion patients, and sample control patients. And during doing this t-test, I found out that there were differentially expressed genes between all the 3 groups. By differentially expressed genes, I mean that they showed genes that were upregulated or specifically downregulated. Later on, doing exploratory analysis, I found out that the variation between control gray matter and gray matter lesion group was the highest. And hence, I downstreamed my analysis for that comparative analysis for gray matter lesions and control gray matter group. Later on, after doing that, I did an HR pipeline analysis, which showed me differentially expressed genes between gray matter lesions and control gray matter. Later on, after elucidating the differentially expressed genes, I did a pathway analysis, which showed the involvement of those differentially expressed genes into different biological pathways. Again, I did a PCA and then further literature review in order to conclude my studies. So for the exploratory analysis, you can see the PCA I did over here. So the first PCA shows the variance among control gray matter and gray matter lesions. The second one shows the variance among normal appearing gray matter and gray matter lesion samples. Similarly, the third shows between normal appearing gray matter and control gray matter, and the fourth shows between all of the three. It is very evident in the PCA analysis that the most variation was seen between group 1, that is control gray matter and gray matter lesion. And that is the reason why I downstreamed my analysis and concentrated only on this group, because they showed me the maximum variance at the transcriptomics level. Moving on to the downstream analysis, that is the comparative analysis. The first thing I did was the differentially expression genes. So I wanted to find out what are the differentially expressed genes between control samples and the patients with gray matter lesions in multiple sclerosis. So I uploaded my RNA-seq sample table on the tBioInfo server pipeline. And after uploading that, I got differentially expressed genes. The values I entered for my differential expression gene analysis was p-value less than 0.05 and log2 fold change plus or minus 0.5. To my amazement, I found 16,740 genes that were significantly upregulated in gray matter lesion samples, whereas 15,960 genes were downregulated. In order, this were very larger results to comply, and hence I decided to move further downstream analysis, and hence I took only top 700 genes for my further analysis. So over here you can see the analysis pathways and whatever the values I took. In order to be double sure for my comparative analysis that the variation is okay or not, I performed a heat map and a hierarchical clustering. In figure number 6, you can see the results of the hierarchical clustering, and it is very evident that control gray matters and the gray matter lesions form distinct clusters, which indicate that there is a variation between these two groups. At the gene level, something is separating gray matter lesions and control gray matter group. Further on, I wanted to see what are the upregulated and the downregulated gene in a pictorial presentation. So I did a heat map. The heat map clearly shows that the significant genes in the lesions, that is gray matter lesions, are upregulated, and same genes are downregulated in the control samples. Hence, the comparative analysis for heat map and hierarchical clustering gave me a thumbs up that there is something at the gene level that separates the gray matter lesion samples and the control gray matter samples. I further on went ahead and did a pathway analysis using Enricher platform. So for the pathway analysis, I segregated my differentially expressed genes and then uploaded on the Enricher platform. Using that, I got the involvement of specific upregulated or downregulated genes in specific biological pathways that were affecting the patients of multiple sclerosis. For the upregulated pathways, one of the most affected pathways was the hematopoietic stem cell lineages pathways. And for the downregulated pathways, one of the most affected one was the olfactory transduction pathway. There it clicked me that in my differentially expressed genes, there were many protein coding genes that were responsible for the sensing of olfactory dysfunction into our body. Also, there were many genes that were responsible for the production of protein kinase A. How protein kinase A is related to this study, we will see further. So what I did was, again, from my significantly expressed genes, I took out all the olfactory receptor genes and plotted them in contrast with the control samples. In my figure number 10, you can see that I have plotted a bar plot of those particularly downregulated olfactory receptor gene levels along with the control samples, and it is clearly very evident that olfactory receptor gene levels are significantly downregulated in multiple sclerotic patients when compared to the normal healthy individuals. This gives us a thumbs up saying that multiple sclerotic patients suffer from an olfactory dysfunction pathway, which was already mentioned in literature review. But what was not mentioned in already studied research was the 12 genes that we found that were responsible for the olfactory dysfunction. Research have not been able to prove till date that what genes causes the olfactory dysfunction in patients of multiple sclerosis. But however, in my study, we were able to portray the top 12 genes that were responsible for olfactory dysfunction. So these genes could be further used as a therapeutic biomarkers or predictive biomarkers in order to treat the olfactory systems in the patients of multiple sclerosis. Another important result that I got into my study was the association of protein kinase A, hematopoietic stem cell lineage pathway, along with multiple sclerosis patients. Now we know that nucleus accumbens is the place where protein kinase A is present, and the regulation of protein kinase A is dependent upon the dopamine receptors. But since the protein kinase A levels was highly downregulated in patients of multiple sclerosis, as per our study, a close association over here can be made between protein kinase A, dopamine, and multiple sclerosis. Dopamine is usually associated with cognitive functions and happiness and mood swings and etc. But since the levels of protein kinase A goes down in patients of multiple sclerosis, dopamine receptors automatically go down and there is lack of production of dopamine. Due to this, there is loss of cognitive functions and loss of impairment. So here we made a possible association between protein kinase A and the function and the cognitive loss in multiple sclerosis. Talking about the conclusions, our study was successfully be able to study the gene expression studies between normal samples and the comparative multiple sclerotic gray matter lesion samples. Not only this, but we were also successful in identifying 12 novel olfactory receptor genes, which was an important outcome of the study because these genes regulated olfactory dysfunctions in patients of multiple sclerosis. We were also able to find out an association between protein kinase A and also the cognitive loss and impairments in multiple sclerosis. Also, I would like to tell that much work is needed in future in order to assess the precise effects of T cell pathways and T cells to multiple sclerosis. And in further studies, there were some pathways like oxidative phosphorylation and ribosomal production pathways that were severely affected. So I hope to analyze these pathways much further in my further research in order to analyze the association of them with the multiple sclerosis. In the purple table over here, you can see the top 12 olfactory genes pathways that we have found in the study along with their functions. You can see that all of those functions in the olfactory systems also. And I would like to thank my mentors who were stood by my side and made this a possible outcome. So this was all about my project and my poster. Thank you so much.